Okay, so we're going to be talking about cell membranes and how they let certain substances in and out. So if all these vocabulary words seem difficult, they can be a little challenging. We're going to break them apart in this PowerPoint. Make sure you're doing the definitions. Make sure you're bringing them in either on Monday or Thursday, depending on what cohort you are. And you might want to draw little pictures if you need it as a reference when we're taking maybe possibly an open note quiz or something along the lines in class. So you definitely want to be prepared for this. These are challenging words. So when we talk about cell transport, there's two main types. So you have to keep these set apart. One is passive transport, where no energy is needed. It happens naturally. So the cell doesn't have to be alive for this even to take place. And then active transport, where the cell does need energy and requires that in order for those actions to take place. So let's break those apart. So the first example is going to be passive transport diffusion. So diffusion is, the definition is going from an area of high concentration. So I'm going to represent that by H, going to area of low concentration. So the universe works this way. It goes towards entropy, chaos. Imagine your room. Does it become more highly organized? Or does it naturally become more messy? So the way the universe works is it goes towards a state of entropy where it becomes more chaotic. So that's how molecules move. They move towards distribution, right, or equilibrium, or they're spread out, right? Just like your clothes might be organized in a closet, and if you don't expend energy, all those clothes might spread out over time. So that's how molecules are going to work. Right, so diffusion is going to be, examples are going to be small molecules, carbon dioxide, water, oxygen, and they're going to go with their concentration gradient. So once they've reached a concentration gradient, we call something equilibrium, and that's where the molecules are spread apart equally. So here we'll do a quick demo in class. The molecules are going to move from an area of high to an area of low concentration. So some examples that are going to diffuse through the cell membrane naturally, again, no energy is needed, is there going to be small things like gases, like carbon dioxide or oxygen, or even fat-soluble substances can pass right through the cell membrane. Remember, the cell membrane is a fat layer, and then these are fats. These are fats right here, and these are phosphates. But again, it's like an oily layer that's separating the outside of the environment from the inside of the environment. So other things that can pass through are water, alcohol is another example that can pass right through, and then larger molecules like glucose or amino acids or even charged particles just can't pass through the cell membrane or through diffusion, through the process of simple diffusion. Another example of passive transport is something called osmosis, where water is going to be moving in and out of the cell. So it seems to always be that they want you to understand the concept that an area of high water concentration is going to go to an area of low water concentration. But the reason why you had that change from an area of high water amount right over here versus an area of low water amount is based on what's dissolved in the actual solution itself. So when we say an area of high water, well, that's because there's low solute level. The reason why there's less water here is because there's more solute. All right, so let's see what those terms mean. Solvent is usually the water that's going to dissolve the substances, and solute is either going to be solids or going to be gases that's actually dissolved in the substance. So when we're using the word solvent and solute, we're talking about a solution, right? So going back, this is an example of a solution. So we're always talking about the water concentration in comparison to another. So let's just see what that means in an example. So if we have our solution, right? They're telling you it's 10% salt. So they assume that you know that this is a solution, right? And they should tell you, like in any of the questions, that they are solutions, right? So you know then that 90% is going to be water, and the other 10% is going to be salt. Right, so how did I know? Because obviously this is going to equal 100, so that's how I get my simple math. So here, the same thing here. So they're telling you how much solute is in there, which is salt, and then 90% water. So if there's equal amount of water on either side, then the distribution is going to be equal amount. So the area is going to, or the molecules are going to move equally in and out. All right, so you'll actually see no change inside the cell. In this case, you have 98% water. And how do we know that? Well, because we have 2% salt. And here we still have the same. We have our 90% water. So here you have to pay attention. It's not so much the salt concentration, it's the water concentration, because the salt can't pass through. A lot of people think that the reason why the cell is changing is the actual solute is moving. That's not the case. It's the water that's moving, hence the word osmosis, which is the movement of water from an area of high concentration to an area of low concentration. That's the trickiest part here. Is that you have to keep it where you're talking about the water. So once you have that concept, you're good to go. So here we have an area of high concentration of water, an area of low concentration of water. Remember, we got those numbers because we just know that the salt solution is going to equal 100. So an area of high to an area of low is what we're going to see. So actually the cell is going to swell and even possibly burst, which is not a good situation. That's why we have kidneys to remove that extra water, and that's why we go ahead and urinate it out. If it's not a human being with kidneys, it could be a fish, it could be a cell with contractile vacuole. There's lots of different types of organs or organelles that remove extra water or conserve water as well. So here we have the same thing here. We have 30%, so we know it's 70% water, and here we're still keeping the same. Remember, the cell is or should be the same in this case, just trying to show you the same cell. So here we have an area of high water concentration, area of low. So literally what's going to happen is now the water is going to leave the cell. So now the cell is going to shrink and then possibly die from it, okay? So all of these different types of solutions that the cell is in is named different things. So hypertonic is where it's a sugary or salty solution. Hypo is it's a very low solution, and isotonic's just right. So here's the Goldilocks I keep on mentioning in class. Everything in life has to be just right. So that's iso, right? Isotonic is equal concentrations. Hyper is like too hot. Porridge is too hot. Goldilocks doesn't like it. Hypo is too low, meaning it's too cold, and it's not right either. So again, it has to be just right. Otherwise, you have the shrinking and the swelling of cells. So what does that look like? If you have a hypertonic solution, this is showing you where if you did this, if your kidneys weren't working very well, I'm just trying to move this over. If you move that over, I had to move that over, but you could see this, the water is actually leaving the cell, right? Here, isotonic, it's just right. So here they're still showing you movement of water, but it's happening at equal rates. That's why we call it equilibrium. So as much as going in is the same as coming out. And then hypotonic is where there's very little solute concentration, right? And that means the water is going to be going into the cell, and that's going to cause it to possibly burst. If that takes place, it's called plasmolysis, right? So with plants are going to be a little different. They actually like a hypotonic solution the best, and the reason why is that creates a lot of turgor pressure. Turgor pressure is where water is coming in because the plant has access to a lot of water, and it's going to be storing that in the central vacuole. And that's actually going to apply pressure to the cell wall, and then that's going to make the plant stand upright. As it goes towards actually equilibrium, in this case isotonic, it does become a little bit more wilty or wilting, the type of plant. It all depends on the type of plant, but it could possibly wilt. And of course, if it's completely plasmolized, it'll be wilted or even possibly dead. So plants actually like a hypotonic solution. And that's again for that turgor pressure. So those are all examples of passive diffusion. Passive diffusion is where it just happens naturally through the cell membrane. Nothing is really needed at all. It'll just go ahead from an area of high to low. Facilitated diffusion does the same thing. It's still diffusion, and it's still passive. No energy is needed. And it still goes from an area of high concentration to an area of low. But the difference here is that you actually have to be facilitating that diffusion. That's why it's called facilitated diffusion. So facilitate just means to help. So these examples are going to be very either large molecules, and they're not soluble in lipids, so they can't pass through the cell membrane, or they actually need to be carried across. So examples would be like potassium or sodium ions are going to need facilitated diffusion, or even large molecules like glucose are going to actually need to be facilitated across the cell membrane. So again, they need a carrier protein or protein channel. Another type of facilitated diffusion is where you actually have ion channels. These are type of proteins that allow particular ions into or out of the cell. So these are really, really important when we're talking about uh, nerve cells. And we'll talk about these when we talk about nerve cells and action potential. So, and how nerves actually work is that they need to have a particular ion. So you might hear like having Gatorade is really important, or having your electrolytes, or even salt in your diet. And that's very important because these actually allow these channels to be open, so you can allow signals to be passed inside and outside of a cell. So these are really, really important in electrical signals. And we'll talk about those gated ion channels when we talk about nerves. And here you can see just some examples. Here it is closed. And again, these pluses stand for ions. So like a sodium ion versus a potassium ion. All right, so now we're getting a little different. Now we're talking about active transport. Active sounds just like it is. It requires energy. So we said diffusion was, whoops, sorry about that. We said active, we said passive was high to low. And so we know that that is going to be this way, right? Going low to high, right? That's diffusion. I'm going to write D for diffusion just because it's pen. Now if we want to go the opposite way, so we're going against the concentration gradient, right? We're going against what the universe says. We're going towards organization, not chaos, right? So that means an area of low concentration is now going to get condensed to an area of high concentration. So if you were to, go up a hill, you'd have to expend energy, where here you might just slide down it passively. So this is going to be active transport. So I always tell students to either, memorize this quick little demo, or a little way so that you can go ahead and get any active passive transport question right on the regions. Okay, so it requires energy, ATP. So sometimes if you see like any ATP, it's a little hard here in any of the examples, right off the bat it's active transport. And even if you don't understand what's going on here, well, again, this is a sodium potassium pump, which is how nerves work. So we'll talk a little bit more about that. But this is, again, active transport. And ATP is always a clear indicator. And again, ion channels and carrier proteins are going to help that stuff be transported, right? So literally it'd be like a trucker, grabbing material and then transporting it to the particular place that it needs to get to, right? So here's sodium potassium pump, which is active transport. This is where a cell membrane is going to transport sodium potassium ions up from a low concentration to an area of high concentration. Okay, and that's going to be an example of primary active transport. So some animals are actually going to need this higher concentration of sodium outside and a higher concentration of potassium inside the cell. We'll talk again about that. That's when we're talking about nerves. But that's really a clear indicator of how our nerves work. And so once we set up that concentration gradient where you have sodium on one side and potassium on the other, it's almost like a battery. So now you have your pluses on one side and you'll have minuses on the other, very much like a battery would be. And then you'll have your secondary active transport, where that transport proteins by the gradient created from the primary. So what's going to happen is now, once you set up that concentration gradient and you trigger a nerve, you're going to go ahead and you're going to release those proteins based on their concentration gradient. So that's why a nerve needs to have two actual signals in order for it to work. The last one is movement with vesicles. Again, this requires energy. It's active transport. So these are usually larger molecules. And so just remember, exo is leaving, so it's exiting, and endo is coming in. So when a plant or an animal cell is taking in large substances, it's called endocytosis. And when it's getting rid of larger molecules, it's called exocytosis. And this always is going to require energy. So these are just examples of endocytosis. Again, that's active. Here it's highlighted yellow. Some two examples of endocytosis is always in endo or exo, there's actually a vesicle surrounding, and that's actually a pinching off of the cell membrane. So one example would be pinocytosis. So pinocytosis here, it's almost like they call it cell drinking, and this is where large molecules are taken in, more of like in a fluid state. And phagocytosis is going to be very, very large substances, so almost like a single cell, like bacteria. And that's going to be phagocytosis, so transport of large, large particles or even whole cells. Phagocytosis is, again, example would be a phagocytosis would, like if a cell was eating bacteria or viruses, that would be an example of phagocytosis. The other one is going to be hormone or cell-mediated response. So that's literally where, let's say, a hormone comes in, and we'll talk about this when we talk about the nervous system and endocrine system. So actually what happens is it clicks onto a receptor, and then it pinches off the cell membrane, and then that information is taken into the cell, and in this case, it's showing you that an antibody is being made. So again, this can't happen unless the actual receptor is allowing that material to come in. So that's called cell-mediated um, uh, response. Okay, so exocytosis is where materials, that's a little bit more easily explained. So literally, again, it's like this pinching off of the cell membrane, and literally materials are coming outside of these, in this case, of the cell. So again, this process allows excess waste, toxins to come outside. Okay, that's it. And I will see you in class.